after the Hodgkin-Huxley hell, we're going to just take a breather and just talk about the third sort of main conceptual chunk of neurophysiology and neurons communicating. So we started off with the resting potential. And so the resting potential, mainly due to potassium, you've got these leak channels. There's more potassium inside, a little bit of it leaks out, the inside gets negative. That was the resting potential. And then it eventually reaches a balance because the inside's negative and it will attract the potassiums back in. So that was the resting potential. Then we talked about the voltage-gated channels. So those are channels that actually change their open or closed state on the basis of the voltage across the membrane, and those were, we talked about the voltage-gated fast sodium channel and then the voltage-gated delayed rectifier potassium channel that sort of puts things back. And so today, the third main part is, let's talk about postsynaptic potentials. And postsynaptic potentials are caused by channels that open up when a neurotransmitter hits them. So these are called neurotransmitter-gated channels. So they're as opposed to voltage-gated channels. So it turns out they're genetically related to voltage-gated channels. It's just that they've got a different apparatus for opening the channel. And where are these channels typically located? Well, they're typically located at a synapse. And so what you've undoubtedly seen pictures of synapses that look like this. Little ball like that, little synaptic bouton. What do they actually look like? So if you go to the MRI center, have a nice diagram of what an actual axon terminal looks like. And so you'll see there's like a big axon, myelinated axon coming in, and then it breaks up into a whole bunch of little mini axons, and the spike sort of splits up into, hundreds of little spikelets. And then there's like 3,500 little synapses all over this. Each one of those guys, so this whole thing is an axon terminal. But each one of these guys right here, corresponds to this little synaptic bouton. So you often get the feeling like there's a couple of these. There's like 5,000 coming off of one axon, so there's a lot of them. And so what are they stuck onto? So I'm drawing the synapse. This space in here is the synaptic cleft in between the two cells. So this guy is, a synaptic bouton, little synaptic French button. And this guy's a dendrite. Could be a spine, but let's just draw just a flat dendrite. That's a flat dendrite. And this thing is a synapse. And there's actually, I always like to say this, you hear this idea of strengthening synapses. It turns out that's actually, it's not a metaphor. There are little guy wires that hook up this synaptic bouton to the dendrite. And as you strengthen a synapse and make it more effective, it turns out it actually physically gets stronger. So you can show that a stronger synapse that makes a bigger effect on this dendrite over here, if you try to pry it off the dendrite, it's actually stuck onto the dendrite more. So when you're strengthening your synapses, you are physically strengthening your synapses, actually making them stronger. So how does this work? Well, we've got an axon. This is the end of an axon. So we have a spike coming down the pike. So here's a spike coming down the pike like that into the axon. And what does it do when it reaches the synaptic bouton? It causes some calcium to rush in. And calcium is used for signaling all different kinds of things. You know, calcium can do 10,000 things in different cells. It's just one of the things that, you know, an inrush of calcium is going to, you know, in this case, cause a synaptic vesicle to fuse with the membrane. And so calcium is often involved in sort of vesicle fusion type things, but it's also used for a zillion other things, and we'll see it's going to be used for changing the strength of a synapse. It involves calcium also. But this calcium here has to do with how the spike essentially causes neurotransmitter release. And so the voltage change of the spike causes some calcium to rush in. That then causes some neurotransmitter to be released. And so the neurotransmitter is in these little vesicles. And here's one that just fused. So the neurotransmitter is inside here. So, you know, what's, you know, for example, glutamate. There's, you know, for example, glutamate is packed inside there. And so when the calcium rushes in, it causes one of these vesicles to fuse with the membrane. And then what does that do? That then that binds to the channel. And so here's our channel. So here's our channel, and that's the little binding site for the glutamate. So the glutamate will leak out of here and then stick into that little site. So that's a little glue. And when the glutamate sticks to the channel, that's a neurotransmitter-gated channel, it causes the channel to open. And so if this was, say, a sodium channel, it lets in some sodium. So that's basically what causes a postsynaptic. So the meaning of postsynaptic potential is just a potential that's past the synapse. So here's the synapse, so it's a potential in the dendrite caused by the glutamate binding. Now what does that what does that actually look like in voltage if we just opened one of these guys? So if we just opened one of these guys, if here's like 0 millivolts, we might be, say, at minus 70. So here's the membrane potential. If we just opened one of these guys, you get a little blip like that, and it goes down. So how big is that little blip? So this little blip is called an excitatory postsynaptic potential. That's, excitatory PSP. And how big is it? Pretty small, like maybe 1/3 of a millivolt, for example, 0.3 millivolts. This distance here, not very much. And that makes sense because we need a lot of synapses, like 100 or 500 synapses going off almost all at the same time in order for the cell to be activated. So we don't want just one synapse to activate the cell. It's got 10,000 inputs. So that's an excitatory postsynaptic potential. And what does an inhibitory postsynaptic potential look like? Pretty much the same. So here's 0 and minus 70. So you'll be going along at minus 70, and if you trigger off an inhibitory postsynaptic potential from a different kind of channel, you'll get a little dip. The dip is a little bit longer, something like that. So that guy would be, the time course is a little bit longer. So this guy would be an inhibitory IPSP. Inhibitory PSP. So just like in the case of the voltage-gated potassium, those will oftentimes be potassium channels, but they can be chloride channels too. Okay, so that's so now let's just say two things about that. So the first thing is, so if I blow that up, you know, what does it look like? It looks, you know, it looks like that. Of course, everything looks like that. Now, when we drew that graph, so you need to know about this shape of a graph, but there's about like 20 things that have this shape. So so that graph down here looks like that, but what's going on here? It's different than in the case of the voltage-gated channel. So in the voltage-gated channel, what was causing this was the uprising part of it was the fact that the channel was being activated, and then what was causing the down part, that was the channel was still activated, but it was on top of that being inactivated. This separate process of sort of channel blocking that has like a longer time constant. So that was kind of activation and inactivation. So what's going on here is a little different. So what's going on here is we've got, if we sort of blow it up, we've got this, you know, protein like this, and it's got a little receptor site with a very specific shape to it like that. And then that whole thing is attached to a giant channel. And so this guy is the receptor. It's shaped exactly like the thing that's supposed to stick in it. So say here's a glutamate. It's just about exactly the same shape as that glutamate, so that it will stick in there. And when something sticks in there, it will cause the channel to pop open. So that's what's going on, you know, over here is the, you know, receptor binding, you know, binding to receptor. That's what's causing it to go up. But what causes it to go down? It's not inactivation. It's a different thing causing it to go down. So it turns out there's a whole bunch of things that cause it to go down. But in order to understand this, you have to realize that when this guy, you know, is called, you know, the ligand, you know, for example, neurotransmitter. You know, for example, glue. So it turns out this ligand, the little pocket for the ligand cannot be exactly the right shape as the glutamate. And that would be a bad thing. And why is that? It's because then it would really stick. And so when we talked about something like tetrodotoxin, it has exactly the correct shape, so that when it sticks into the channel, it's hard to get out. And if the glutamate pocket was exactly the same shape as the glutamate, that wouldn't be a good thing, because what would happen is it would just stick in there and jam the channels open. And so it turns out it's a subtle distinction, but it's very important that the receptor is ever so slightly different shape than the thing it's supposed to receive. And the reason for that is that way the ligand, or the neurotransmitter, can get out of the receptor, because obviously we want to just sort of signal something, open that guy up, and then we want to clean up and start again. And so, you know, there's a cleanup crew to get rid of, you know, any glutamate that's in the channel. So there's a bunch of guys that come in and like sweep any glutamate out of there. It diffuses out of the, it comes unstuck. Why does it come unstuck? It comes unstuck because it's not exactly the right shape. Close, but not exact. And so it'll stick in there for a little bit, but it's not quite the right shape, and so it comes out. And when it comes out, then the cleanup crew can get it, or it just might diffuse away. So there's a whole bunch of things going on here that creates this sort of same shape as we had before. But it's important conceptually to distinguish like what's going on here. So what's going on here is we're releasing a neurotransmitter. It's sticking into the almost exactly right shaped pocket. It's causing the channel to open up so that something will go through the channel, and then it's getting swept out of there. And so the other thing I always mention, I'll put that off until we talk about the main types of channels. Okay, so that's the basic mechanism of a neurotransmitter-gated channel. Something comes out, sticks to it, causes the channel to open up. Eventually the ligand that's sticking to it diffuses away, gets cleaned out, and so then the channel closes. So that's what causes the channel to close. So there's lots of different kind of channels, but let's just talk about 4 really biggies. And so these 4 really biggies are basically all, well, a lot of them are defined by poisons. And what are poisons? Poisons are typically things like tetrodotoxins that have exactly the right shape of a receptor. And so they just jam into the receptor and they won't come out. And sometimes these are called suicide inhibitors. They just go in there, bam, and they can't get out. And, plants and animals have tried to figure out how to make these in order to keep other plants and animals from eating them. So what's one of the real biggies? And the real biggies is called the AMPA channel. So we'll talk about 4 main postsynaptic potential channels that are neurotransmitter-gated. And so the first one is the AMPA channel. So AMPA it's a small molecule, very poisonous molecule, but it names the channel. So it's the AMPA-glutamate-gated sodium channel. So that channel is, so it's a sodium channel, and so it turns out it's not quite as clean of a sodium channel as the sodium channel that causes the spike, so other stuff can get through it. And so it turns out the reversal potential or the Nernst potential, quote unquote, for this channel is closer to 0 than positive. Like the voltage-gated channel, if you just look at the distribution of sodium inside and out, more outside, less inside, that causes a very positive Nernst potential, like plus 55. But this guy's more closer. It's got some potassium going the other way, and so it's the reversal potential, or the effective Nernst potential for this channel is about 0. It's aiming for about 0 if you open it up. I mean, at rest, obviously it's got a long way to go to 0, so it's definitely going to be up if you're at rest. So this guy is, the excitatory, sodium channel. And so glutamate is the thing that sticks into this channel. It mainly conducts sodium in. The effective Nernst potential is about 0 millivolts, so it's a little leaky, and it's not voltage sensitive. So those are kind of the main properties of it. Now, interesting thing, this is one thing I always say, an interesting thing about the blood-brain barrier is it's so good, you can just eat glutamate. So like MSG flavor enhancer, you can just eat that. That's actually the main neurotransmitter, one of the main excitatory neurotransmitters in your brain. If you actually injected a little glutamate directly into your brain, all hell would break loose. It would just, you get sort of seizure-like activity, sort of, frying the brain. But you can actually eat that stuff, and your blood-brain barrier will keep it out of your brain. It's kind of, I compare it to like, pouring salt water onto your laptop. Pour a conductive fluid onto your laptop, and the laptop just laughs it off. So that's no problem. Keep going. So you might get a little bit of a little red nose or something, maybe a slight headache, but basically you're okay, even though you just ate pure neurotransmitter that all your neurons in your brain are using to signify excitation to each other. So the other thing I often mention is like AMPA. What's that? Well, and if you ever heard of Roundup, glyphosate. Glyphosate is something used to kill weeds. So a lot of people sort of use it out in their front lawn to get rid of the dandelions. And it's heavily used in the food industry, not just for killing weeds. They turned out it's often used to dry out crops in the field. They dump tons and tons of glyphosate on the fields in order to quickly dry out the crop so they can harvest it. And there's a lot of talk about maybe it's not so good to eat glyphosate. But it turns out AMPA is one of the breakdown products of glyphosate. So I always tell the students, if you put this on your front lawn, do not go out and lick the front lawn, because that stuff is much more nasty than glutamate. You can eat glutamate, you don't want to eat AMPA if you can avoid it. Okay, so that's the AMPA-glutamate-gated channel, main excitatory, non-voltage sensitive, neurotransmitter-gated channel that causes excitation, in the brain. So what's another one? So another one is called the NMDA channel. NMDA. So this is also glutamate-gated. And it's not just a sodium channel, but it's also a calcium channel. And it turns out the calcium part is going to be important. So this is another major channel. So NMDA is it's N-methyl-D-aspartate, but whatever, it's just NMDA, and it's something that sticks in the glutamate channel in the NMDA channel, and the NMDA sticks in the NMDA channel. So this guy is the learning channel. So this is the one that's learning. This is the Hebbian channel. We'll talk about this later. And so how does this sort of Hebbian later in the second half? So how does this work? Well, this guy is voltage also. It's sensitive. It requires glutamate, but it's also voltage gated. So it's both. So this is, if you want to know what life without NMDA channels is like, you can try ketamine. So ketamine is something that basically blocks NMDA channels. Small amounts of ketamine will block NMDA channels that are involved in pain. So it's a good painkiller. If you have enough of this, it will lower the excitation enough to cause loss of consciousness. But that basically specifically goes to NMDA channels and shuts them down. So those are some main excitatory guys. So these guys are excitatory. And then we have two biggies for inhibitory. And the first one is called GABA, the GABAb channel. This is often written like that. So the GABAb channel is a potassium channel. So you'd expect this guy to be an inhibitory channel, and so this guy is called the hyperpolarizing inhibitory channel. Hyperpolarizing inhibitory channel. And this guy is, something like minus 100 millivolts, minus 90, minus 100 millivolts. It's aiming definitely below rest. So rest is minus 70 millivolts. And so this if you open this one, it will drive you down definitely below rest. It's slightly slower than AMPA. the inhibition always tends to be slightly slower, or can often be slightly slower than the excitation, like in the spike case, but the inhibition also tends to spread out spatially a little bit more than the excitation, and those are just sort of good rules of thumb in order to keep activity from just going completely crazy. So that's the GABAb potassium inhibitory channel. And then there's another one called the GABAa chloride channel. And this is another extremely important inhibitory channel all over the place. And this guy is sometimes called the shunting inhibitory channel. I'll try to explain what we mean by that. So if we look at the reversal potential for chloride, it's about equal to rest. So think about what that implies. So that means that it's trying to drive you to the resting. It's not trying to drive you below the resting, it's just trying to drive you to the resting. So that means if you open this channel at rest, nothing happens. Yeah, open at rest, no effect. So you can see why this is a good channel, because this channel is like, let's put everything in order, put everything back to normal. So the current can go either way through this channel. So if you're below rest, so if you're at like minus 90, like somebody opened up a GABAb channel, and you open up the GABAa channel, the GABAa channel will raise the membrane potential and get it back to rest. So it's a very sort of put everything back to normal kind of channel. So that's the other main inhibitory channel. And alcohol affects this one. So, how does alcohol affect this one? It basically causes it, if you drink a lot of alcohol, it gets into the membrane and slightly changes the nature of the bilayer lipid membrane and causes these channels to stick open a little bit more. And so alcohol is inhibitory. It just turns up the inhibition. You can sort of think of it as just turning down the volume. If you drink a lot, if you drink enough alcohol, the volume goes way down and you pass out or die, right? So, and this is, I think I might have mentioned the last time or 2 times ago, but this is a great example of the ways in which these networks of neurotransmitters adapt in order to always keep the system within a particular homeostatic range. And so if you drink huge amounts of alcohol, so if I drink a 1/5 of gin, I'll completely pass out. It would just completely knock me out. But if I were to drink like just a glass of gin and then 2 glasses of gin and 3 glasses of gin, do it for about 2 to 3 months, I could get up to, if I just routinely drank heavily like that, I could get up to drinking like a whole bottle of gin and still be standing. And that's what happens with alcoholics. They can actually, but just think about what that requires. In order to still be standing with these GABA channels jacked up massively, you've got to do something. Either get rid of some of the GABA channels or turn up some of the glutamate channels or the NMDA channels, turn down the hyperpolarizing channels. The whole meshwork of things adjusts in order to allow you to drink a 1/5 of gin a day and still be operational. Of course, if you then stop drinking a 1/5 of gin a day, all hell is going to break loose the other way, because you'll be way overexcited, and so you can get an actual seizure because your system has adjusted. So that will sort of make you remember the GABAa chloride channels. This is benzodiazepines mess with this. The sleep aids like Lunesta. Lunesta is like instant drunk. You take that, it just slams into your GABA-A channels and really turns them on, and it just hopefully it makes you go to sleep. But sometimes you get into this weird state where you get in your car and drive out to the bar, and then you wake up in the bar. But yeah, probably better to just drink a little less coffee in the morning, then you'll get to sleep better. Okay, so now let's turn over and it's one last thing. We should just sort of quickly describe something that you might see. Like, this is one way of classifying the channels that sort of classifies them by their main function, their neurotransmitter and their main function. But we could also classify them by their actual structure, by their actual proteins. And so we'll talk about three main classes of channels. And so there's one class that's called 4+1 channel. It has to do with those alpha helices going through the membrane. And these are called nicotinic acetylcholine. That's acetylcholine receptors. Okay, so there's and an example are the synapses in muscles. So the actual channels at the synapses in muscles. So these would be channels on the muscle side that are picking up the acetylcholine instead of glutamate. There's some serotonin related, these 5-hydroxytryptamine channel number 3. That's another example. But another example is the GABA-A channel. So the GABA-A channel is one of these. And so you can see that the particular structural group that the channel falls under doesn't define its function. This is kind of like more of a functional approach. What is the main function of the channel? This is like, what is the structure of the channel? This is a 4+1 channel. It's got 4 subunits of one thing and 1 unit of another different thing that form the pore that goes through the membrane. But then there's another set called 4G ionotropic. I didn't make up the name. Okay, so that's so there's a whole bunch of non-NMDA channels, and then there's the NMDA channel. So, and that's one that we talked about. So that's that guy. That's that guy right here. And then finally, there are ones called G protein coupled receptors. And those include the AMPA channel and also the GABA-A channel. So you can see you can be inhibitory or excitatory. It just has to do with the fine details of how you've messed with the pore in the channel. But all these channels have a similar overall structure, but completely different function. So here's the GABA. Oh no, that's not right. I got GABA-A twice. Man, I wonder how long that one has been there. Can't remember which one is which. Let's see. It's got to be here somewhere. Okay, well, I can't remember whether this guy was GABAb or GABA-A. It might be backwards. I'll properly look it up and bring it next time. GABAb. Okay, got it. Okay, so that's one that we talked about. That's the potassium channel. Okay, we talked about the AMPA. That was the glutamate. Okay, very good. Okay, so those, like the structural classification, functional classification. Okay, so now let's go over to changes in the strength of synapses. And so obviously one of the things that we're interested in is actual learning and memory. And learning and memory imply that somehow we've altered the synaptic strength. And the initial investigations into this were done using brain slices. And so brain slices, why would you want to slice the brain? One of the big things about a brain slice is it's obviously just a slice of a brain, but it doesn't move. And so it turns out if you have a living slice, thin slice of the brain, you can keep it alive and do things that are completely impossible in an awake or even anesthetized animal because of microscopic movements of things. And so there's all different sorts of extremely high-tech things you can do. So how do you get a brain slice? You very quickly take the brain out. So we're talking like in 60 seconds. And then you make a very thin slice of the brain, put it in a dish, and then perfuse fluid over it to keep it alive, and then you can record and do all different kinds of very high-resolution studies. And so these studies were initially done many years ago, like 30 years ago, and they figured out something called, LTP for long-term potentiation. So potentiation means, it's like potentiation, but it means long-term increase. So long-term potentiation meaning long-term positive increase in the strength of a synapse. And what is the most basic setup is to get a piece of the hippocampus. And in the hippocampus, you can find places where there's a whole bunch of fibers coming in and making synapses on a hippocampal cell. And you can get this whole structure in a slice. Now, obviously, the slice is pretty bizarre because you cut off all the other connections to the slice. And so the first thing that happens in a slice is there's absolutely zero activity. Normally, if you record from the brain, there's some stuff. There's always some background activity. But once you cut a slice, you cut off all these inputs, and it's completely silent. So that's kind of nice for trying to measure small currents and things like that. But it's obviously pretty abnormal. But what's the basic LTP experiment? The basic LTP experiment is to put a big electrode here and stimulate a whole bunch of these, sort of non-specifically, just sort of stimulate a whole bunch of these axons coming in, and then put a fine microelectrode pipette into here and record inside the cell. So it's the most basic setup. We've got some inputs. They're synapsing on a cell. We record from the cell, stimulate the inputs. And the basic LTP experiment is very straightforward. It's simply start at -70 and then do a fixed small stim. So what are we doing here? So we're taking the stimulating electrode and do a fixed amount of stimulation, small stim. And then we can do it a bunch of times and sort of measure like what the amplitude is. And so we get a certain amplitude of that. Here's the amplitude of that postsynaptic potential. So this is, so fixed small stem, measure, height, PSP. So measure the height of the postsynaptic potential. Then what we do is we slam it to the ground, and that's called a tetanus. And it's just named after, so this is, it's named after like tetanus. You get tetanus toxin and it causes all your muscles to contract, very strongly to contract. So that's what a tetanus is. So we do a very strong, so this is, strong stim. And that is called a tetanus. You can just imagine how people figure this out. They say like, wait, let's turn it up to 12, see what happens. So we do a strong stim, and then we wait some time. You could wait, you could just test it immediately, but you could wait like an hour, and then do the test again with the small stimulus, and it's bigger. So then just do this test again, retest. So that's the basic, this is basic LTP observation. Now, when you look at this, it's kind of like, yeah, okay. And so what is the long part? The long part just means that this lasts for at least a couple hours. So, as far as long, well, that's pretty long, So I mean, just 1 second after doing this, you can see that already there's an increase in the strength. And then if you do it, test like 3 hours later, the increase in strength is still there. So when you first look at the basic observation, you say, well, this doesn't look like much, it's just, how are you going to get anything out of that? But using techniques that you can do in slices, people have figured out in detail how this is working and how it requires the NMDA channel. And so now let's draw a synapse again. So here's a synapse again. And now we're going to draw 2 channels. So here's our dendrite. And so first we have an AMPA channel. So here's our AMPA channel. This guy's the AMPA channel. So it turns out that LTP involves sort of an interplay between the AMPA channel and the NMDA channel. And here's the NMDA channel. So remember, they're both glutamate-gated channels. So glutamate is the neurotransmitter for both of these. So here's some glutamate, some glue in there that just got released. And so that will go out to the AMPA channel, and it will also go out to the NMDA channel. So when it goes to the AMPA channel, it's going to, cause some sodium to come in. When it goes to the NMDA channel, if the cell is at rest, the channel is going to be blocked. So if the cell is at rest, nothing will go through the NMDA channel. If the cell has been, so this is the, I forgot to label this. This guy's the dendrite over here. So this is, synaptic bouton. And this guy's dendrite. So synaptic bouton. So one of the sort of roadblocks in understanding this is it's an important point. It's often kind of left out. So the glutamate at one synapse, so the glutamate release at one synapse is not enough. This is critical. Not enough to unblock the NMDA channels. So what blocks the NMDA channels? There's basically a magnesium, so Mg blocks. So when at resting potential, blocks NMDA at resting potential. So it's a different mechanism than the mechanism of the voltage-gated channel inactivation, because that was like a little piece of the channel that kind of like jammed into the channel. This is different. This is just an ion sticks in the channel at resting potential, and an ion just blocks it. And so the amount of depolarization caused by the glutamate hitting the AMPA channel here is not enough. That's a critical point. And so what does that imply? So the implication of this, the critical implication of this is that other synapses must help depolarize. To unblock. So you won't get anything going through the NMDA channel unless some other guys somewhere else on the dendrite have also opened up AMPA channels and gotten some depolarization going, so that this guy can experience enough depolarization to unblock himself, and then allow the glutamate to open the channel. So that's basically what's been figured out. So what happens? So in this case, sodium comes in. In this case, you get some sodium. There's different NMDA channels. Some of them can block more or less sodium, but critically, this one also brings in some calcium. And then a whole bunch of things happen. And if we had all day, and Marty had actually read about these, there's a whole cascade of phosphorylation that's created by the calcium, because calcium is just like a massively important internal signal. And so this eventually ends up, amazingly, messing with something called nitric oxide synthase. So it turns out so what is nitric oxide? Nitric oxide, I always say this, nitric oxide is that rank smell that comes out of I was in London for about 10 years, it comes out of the back of a very poorly constructed diesel engine in a black taxi in London. It's that stink that sort of comes out. It's a poisonous gas, and it'll kill you. But you've got an enzyme that actually generates a tiny little puff of that poisonous gas, and it's poison if you just take a whole bunch of it. It's obviously not poison for the cell, because that's how the learning works. And so that thing will actually generate some nitric oxide, NO. That's nitric oxide. And the nitric oxide will diffuse out of the cell back into the synapse, and it will actually increase the probability of release. But there's also feedbacks from the calcium onto the AMPA channel, and it turns out, and so nitric oxide is used all over the body. Nitric oxide is very important for dilating blood vessels. So it's used to signal when you need to open up a blood vessel and get more blood flow. Nitric oxide is. That's why they used to give people nitroglycerin for heart disease, because if you have heart disease and your vessels are not in good shape, when the signal comes to make nitric oxide and then respond to the nitric oxide, you don't respond to it quickly enough. And so if you take nitroglycerin, that will break down into nitric oxide and cause your vessels to dilate and cause less heart pain, because then you'll get enough oxygen to your heart. So that's used all over, but it's definitely used in LTP. And what are the basic things I always ask on the test? Well, so this is kind of a Hebbian mechanism. And what is Hebbian mechanism is detect pre-post correlation. So how does this channel do that? Well, it detects the presynaptic by glutamate. So it requires glutamate in order to open the channel, and that indicates, so, presynaptic, activity detected by depolarization, detected by glutamate. Okay, so how does it detect the postsynaptic? Postsynaptic, detect by depolarization, because it requires both of those. It's an AND gate, AND gate. So it requires both glue and dipole. And then when it opens up, it lets the calcium in. But it turns out, this is a critical point, it doesn't require the NMDA channel to express this. So this is a critical point. Can block NMDA after LTP still there. So it's not actually expressing the strengthened synapse. That has to somehow be expressed through the AMPA channels or by messing with the vesicle fusion. So we could either, we could just tell the cell make more AMPA channels so that the same amount of glutamate will give you a bigger burst, or we could say increase the probability of release so that when a spike comes down, they'll be more likely to fuse a vesicle. Either one of those or both at the same time could do it. But none of those things require the NMDA channel. So the NMDA channel is only required to detect this correlation, which it does by requiring depolarization to unblock it together with the presence of glutamate. So you need glutamate plus depolarization in this guy, and that depolarization has to come from other cells, not just the present synapse. But if those two things are present, depolarization indicating this guy's active, glutamate indicating this guy's active, then you'll let in some calcium. The calcium will do its thing, and then afterwards, 1 second later, you'll now have a strengthened synapse, which is sort of permanent, just permanently jacked it up. So the last negative 2 minutes, there's one problem you might have noticed, which was, how do the synapses ever go down? You just your brain would just get full, and everybody would just keep turning up synapses. But it turns out when people did more careful experiments, they discovered there's actually a curve of synaptic strengthening that looks something like this. So what I'm graphing is the amount of synaptic strengthening or weakening. And so this is this is, increase synaptic strength. So it turns out if you're going, if the time, so this thing is called spike timing dependent plasticity. So if the postsynaptic spike just after presynaptic input. So this is postsynaptic spike just after, then you increase your synaptic strength. And so what kind of situation is that? So and what is the timing? The timing has to be surprisingly accurate. This is about 100 milliseconds here. So basically, the postsynaptic spike has to occur within, say, 20 milliseconds or 10, 15 milliseconds of the presynaptic input. So the presynaptic input happens, and then this guy has to actually spike almost immediately, and then you'll strengthen the synapse. On the other hand, if you just miss, so this is just missed. If you just miss, so this is postsynaptic spike, just before presynaptic input, then you go down. So think about this. It's kind of, and if you're off by like 100 milliseconds, nothing happens. So, if a spike comes in, and then, or like 100 milliseconds is like that's about 100 milliseconds, like that. So if a spike comes in anywhere more than 100 millisecond gap, nothing happens. It only has an effect if a spike comes in and then immediately afterwards this guy spikes. And so what's the way of thinking about that? You can sort of think about this as a method of detecting that this guy has helped cause a spike. So you can sort of think of this as a causality detector. So if this synapse helped cause this guy spike, then you get rewarded. If this synapse just missed help causing this guy spike, it was off, it essentially, the spike happened before the input came in, that's what this means on this side, then you get penalized. And if you're off by more than 1/10 of a second, either way, nothing happens. So that's spike, spike timing dependent plasticity, and that's our general sort of, current understanding of what is one of the main ways that you learn things, by messing with your NMDA channels, letting in calcium that has the effect of somehow operating on the AMPA channels or on the probability of release, and it makes the synapse stronger or weaker in case you just missed helping cause a spike. Okay, David, I think we'll end my way over recording there.